Good morning, everyone. We're going to start the webinar here in just a moment. We're having people join, so give us a couple minutes and we'll begin the presentation. Let's just give this one more minute, folks, for people still joining the call, and then we'll start the webinar here. So just one more moment, we'll begin. All right, guys, we're going to go ahead and get started. Good day, everyone. Welcome to today's Nikon webinar hosted by Bio. My name is Frey. I'm the National Business Development Director for Bio and the Bio Business Solutions Program. The Bio Business Solutions Program is a cost savings program for Bio members and endorsing state affiliate members. There's a total of 17 individual supplier partners ranging from lab supplies, office supplies, shipping, insurance, corporate credit card, and other products and services. In 2019, over 4,500 individual member companies saved over $500 million on the Bio Business Solutions program. Today, one of our 17 Bio Business Solutions supplier partners, Nikon, will present on advances in imaging technology for research and development, screening, and quality assurance. Our guest speaker today from Nikon is Jeff Bylund. Jeff is a North American Applications Manager for Stem Cell and Regenerative Medicine. Jeff has been with Nikon since September 2015. Jeff has a bachelor's degree of science and chemistry from the University of Utah and a PhD in pharmacology, cell and developmental biology from Vanderbilt University School of Medicine. Some fun facts about Jeff is he enjoys hiking, camping, mountain biking, and speaks fluent Japanese. Before we begin, let me advise everybody on the webinar, there is a question and chat feature that will enable you to type in your questions. We will address the questions at the end of Jeff's presentation. So now let me turn over the webinar to Jeff to begin. Jeff?
Thank you, Peter. I appreciate that, and I'm excited to be here. And Nikon is, of course, very excited to be part of Bio Business Solutions. It's a great program, and we really have enjoyed our relationship. And also the way that Bio has connected us with so many great customers, researchers, and partners over the course of our work together. So thank you very much. And thank you to all the attendees. I'm glad you could be here. This is something that I'm excited about. This is a topic that I find quite interesting, and I hope that you'll be able to find some interest with me. And as I present, the goal isn't to be a download of information, but to be somewhat of a discussion. Like Peter said, we have the opportunity to submit questions, so please feel free and we'll get to those as we can. Today I'll be talking about imaging applications both on the research and development side, but also on the screening and quality assurance side. Imaging as a general application has been around for a very long time, but I want to focus today. I could speak for days about this stuff, and I might be the only one that likes that. But to focus for today's discussion, I'm going to talk about microscopy and R&D and focus on phenotypic assays and then some emerging model systems within those phenotypic assays. And then I'm going to switch over a little bit and talk about what we do with microscopy after the discovery phase, after we've found our therapeutic, especially in an age where therapeutics now are cells or tissues that are being engineered, where the therapeutic itself becomes complex, and so the system for analyzing its quality should be equally complex, or at least be able to grab rich information that can be used to quickly make decisions about lot release, potency, these kinds of things. So to start out with phenotypic screening, I'd like to introduce the concept. So in phenotypic screening, what we're doing is we're just connecting observable traits, the phenotype, with a specific pathology or physiological response that identifies disease-relevant parameters. And then we're using those parameters to test for therapeutic agents, such as small molecules, peptides, biologicals, whatever it is that constitute a specific effect within your model. That's the screening part, right? So we have a phenotype that's observable, that can be affected by some kind of a therapeutic agent or genetic variation, and you put those together and you have a phenotypic screen. Now, this is actually something that's been done for a long time, and in fact, it has its history in doing in vivo screens. This is a paper from 1910 from Barger and Dale, where they used a decerebrated cat to look at changes in blood pressure in response to different amine compounds. And this was kind of the bedrock for pharmacology in the old days. You can see here the graph that's shown is a hand-drawn graph of what the measurement meter was showing as they were taking these measurements. And they could take the organism and they could put different compounds through and see how these affected it. Now, of course, this approach has matured over the years, and now modern phenotypic screening is still done in vivo, but the model systems are chosen that are inexpensive, accessible, and ideally should be high content and or high throughput. So there's a certain level of high throughput that can be done with an intact organism. It has its limits, and we'll talk about that when we talk about in vitro phenotypic screening. But it's still done even today in vivo. And so shown here are some example organisms. We have a rodent, rat, mouse, and we have the zebrafish. And each one has pluses and minuses. In the case of the rodents, these are mammals, and so their fidelity to a human system is a little higher, and their physiology is similar, although also different in ways that are important that we'll discuss later. And then you have a fish, a zebrafish here. These are vertebrates, right? So there is some connection. But the nice thing about zebrafish is you can actually look through them. They're somewhat transparent, especially in their early stages. So these are used a lot for developmental screens. But anyway, the point here is that we want to be able to look at organs intact in an intact physiological system, and then see how inputs or stimuli or genetic variations affect these intact organs or organ systems inside of a whole organism. And so we've come a long way from the days when you would hand record what you saw on your meter, of course, and now we can do some pretty amazing things. So here's some examples of some modern in vivo screening techniques, phenotypic screening techniques. One, here on the left, we see the cranial window technique. This is where a researcher can actually access an organ. Shown here is a brain, but we can look at all kinds of organs by creating a window so that we can use a microscope to look at the response of that organ. Here again is the brain to either genetic variations or stimuli from biologicals or small molecules. Or you can use a genetic mouse model to see how a diseased mouse responds to therapeutic agents. And this allows physiological recordings to be taking place, but also allows us to visualize what's happening in the brain during imaging. Or during the experiment. And so it adds a new aspect of spatial and temporal information that we can collect through the imaging. Another example is this fish in a dish. This is a term that's been coined to describe how we can actually take a lot of living organisms, the zebrafish embryos shown here, and use them for screening across multiple conditions, and then use a microscope to visualize what the conditions look like or what effect these different conditions have on the organism. In both cases, we use modern equipment and techniques together to generate the data. So usually when we're talking about microscopy, looking at whole organs inside of an organism, then we're talking about something like a multiphoton system or a confocal system that allows us to peer through thick tissues. And so we can combine the multiphoton with the living organism, and we can extract some very powerful data in terms of microscopy. In the case of the fish in a dish, then we use automated microscopy. On the right-hand side is one shown, one of our systems from Nikon called the Bio Pipeline Live. And the far right-hand side, we have an incubator where we can keep all of the fish and keep them happy and alive. And then there's automation that's built into the system that will automatically take a fish dish and transfer it onto the stage and then conduct an imaging experiment and then put it back into incubated storage until it's time for the next imaging experiment. And the capacity is for dozens of plates that can each be programmed for an imaging experiment. So this gets more towards high content in vivo microscopy, which is really exciting because that's one of the bottlenecks here is, you can usually get high content, which means you get a lot of information per sample, but the high throughput is challenging when you're looking in vivo. But modern technology allows us to kind of overcome that by automating the process. Another addition that makes this, for instance, observing an entire organism rich in information is we can actually look at behavior of the animal in addition to looking at the cellular level behavior on the physiology side. So we can add a camera to the system, for instance, that looks at pupil dilation or muscle twitch or animal behavior. And we can at the same time observe in an organ like the brain what's happening with calcium signaling or neurotransmitters, neuron firing, innervation, these kinds of things. So we can combine animal behavior level information with molecular level information in the same organism, really bringing out the content that can be observed. So this is exciting. And the question is, what does this look like? So I want to share some data with you from each of these systems before we move on to in vitro screening. So shown here is an example of using the multiphoton to do deep brain imaging. The deep brain imaging allows us to look through, shown here on the left side, very, very thick tissues. And we can go pretty deep into the brain. And we can also look at things happening live with a multiphoton setup. So this data here is actually from Zhang and colleagues who published a very nice example of how they could look at particles moving through vasculature, which is shown on the top. The vasculature is outlined in the red here during, in a live animal, or looking at neuron signaling at a very high rate. So this is on, order of milliseconds, watching the response of neurons to stimulus. So it's pretty exciting, the kind of data that can be extracted. And if you can extract this data and then you can analyze it, then suddenly, many more applications in terms of in vivo phenotypic screening become available. You can look at how genetic variations affect neuron signaling, can affect vasculature formation, how leaky the vasculature is. You can then treat these animals with different compounds and then watch how this affects these systems. And you can do all of this, you can look at all of this in an animal, one animal that's mounted on the system. So it's pretty amazing the kinds of data that you can get. And once this is archived, you can always go back and apply new software analysis methods to old imaging data. Imaging data is powerful because it contains a lot of spatial information, and if you do it live, a lot of temporal information that doesn't expire. It can actually be repurposed and reinterrogated over and over again. So this means that one sample, one experiment becomes very rich. This is one of the powerful things about imaging. Another example of this that goes outside of the brain is shown here. So this is an animal that's been anesthetized, but is still living. And we're looking actually at the pancreas and looking at the pancreatic acidi. And it's been stimulated with an agonist. And what we see here is CO2 oscillations, and that's in green in these acids. And we can see the collagen fibers, that's in the blue there. And this again is quite remarkable because we're imaging at a very high frame rate deep into a living organ, and we can see on a cellular level what's happening when stimulation with an agonist occurs. And this is relevant to diabetes research, but this can be done with most organs now in an animal. So it's very exciting. Now, if we switch over to the fish in the dish, this is looking at zebrafish vasculature and angiogenesis. And we can follow this process with labeled cells that show the nascent blood vessels forming as they move across the fish, across the zebrafish. And angiogenesis is a very important process in development and in disease. And this is something that can be screened for using a high-speed confocal system that's been automated. And in this example, you can see the exquisite detail that's being collected. You can see the projections that are being sent out from the nascent blood vessels as they're interrogating the growth factor space to determine their polarity as they move from the basal portion to the upper portion of the zebrafish here. And then you can see them making connections and forming vascular networks. This process can be seen in real time and with high detail, and we can apply modern software analysis techniques, as shown here, then to quantify this process. We can look at the volume of these the vasculature, we can look at the rate of formation, we can look at the rate of projection generation, we can look and measure how much time it takes for these to form. And so not only can we qualitatively watch these processes occur, but having collected the data, now we can go back and use modern imaging techniques to extract quantifiable information. Shown here in blue is just a mask that's been created that follows the movement of the or the growth of the blood vessels over time. Another example of using phenotypic screening to and the zebrafish to in vivo is looking at heartbeat and extracting quantitative data from those heartbeat images. Again, this is live data, and this is looking at different variants of a genetic variants that affect cardiac behavior, cardiac output, and atrial fibrillation. And what can be done is you can image the zebrafish heart, which is actually quite accessible because they're somewhat transparent at certain stages. And we can take the normal heart and create these vector maps on top of the imaging data, and then we can apply those to the normal and the variants and generate quantitative information along these lines that Mueller et al. Were able to do. And it's quite remarkable, both the qualitative data. I mean, we can sit here and look at these really amazing images of organs operating in the different variants, but then we can quantify this using modern imaging analysis. So it's very exciting, but phenotypic screening in vivo only takes you so far. For instance, it's difficult to screen thousands of compounds in a living organism because, number one, setting up something like this is a little bit more complicated, but also just because the practical point of having thousands of fish or thousands of rats or mice set up for this type of analysis becomes prohibitive. And so there has to be another way to screen candidate compounds. And then what you typically do is you move from those candidates into this in vivo screening that I've already shown you to understand mechanistically what's happening. And so that's where in vitro screening comes in. The in vitro screening allows you to culture cells or even organs, but we're going to focus on cells, and then use those for screening. Cells in culture are far cheaper than the entire organism, and there's a lot more control that can be had, and the density of the cells can be higher in terms of sample replicate number, right? So you can put hundreds of different replicates on one plate when we're working with cells. And so over the course of 10, 15 plates, then you're looking at thousands and thousands of different variations that can be tested. And now we're in the realm of doing a phenotypic screen that's truly high content. Before I go on and talk about screening in the context of cells and the in vitro screening, I want to make a distinction a little bit between target-based and truly phenotypic, and talk a little bit about how these go together and how they're kind of crossing over. But traditionally, when people talk about these screens, they talk about, when looking for new therapeutics, for instance, they talk about taking either a target-based approach or the phenotypic-based approach. In the target-based approach, it's kind of a reductionist approach that focuses on components of a mechanism. So it'll be kind of a signaling pathway without the cell, right? So you'll do some biochemical assays to test an enzyme, for instance. Whereas in the phenotypic assay, it's a more holistic approach where you look at these systems in the context of the cellular systems that they're in, or when you're doing it in vivo, the entire organism, right? And it used to be these were mutually exclusive to a certain degree, but more and more with modern technology, we're able to do phenotypic screens that tell us a lot about the target that we're looking at. And so this is something that's very exciting, and kind of the lines are blurring a little bit. So if we go back and talk about phenotypic screening using cells, then we really have to talk about some of the early folks who kind of commercialized this idea. And this goes back to a concept, a term that was coined called Salomemix. And there was actually a company called Salomemix that did this on a contract basis and generated devices that were used for this kind of phenotypic screening. And what they found was that they could create a multi-parameter assay that could generate 10 to the 8th individual physiological cellular measurements in less than 1 day. So this is what this concept really brought phenotypic screening from kind of the in vivo, very low scale, low content realm into the high content realm, where now we can take, 10 to the 8th individual physiological measurements. That's a lot of data in less than one day. That's not much time, right? So suddenly now we can extract a lot of information in a short amount of time. You'll notice this was back in 2003. This is a term that came up called Salomemix. But this has been refined over the years, and it's become a bedrock for a lot of R&D processes and programs. So what does this look like from a practical perspective? Well, you can look at cells that are fixed, shown here on the left, or you can look at live cells. Each one has its advantages. The fixed time point assays, as they're typically called, have the advantage of being a little more simple and less fewer megabytes. So if you're screening more compounds, then you might start here. The live cell, of course, has the advantage of being able to extract that kinetic information, that fourth dimension of time, and not just space in X and Y and Z. So when you're designing these phenotypic screens, you have to decide, based on the trade-offs, do I need to do the fixed assay and screen, a larger library, for instance, or can I go with the live cells and get both, the context of the cell behavior over time? And I'll show you why each of those would be useful here on some subsequent slides. So the simplest approach, and then shown here, are Nikon examples of instruments that are designed to either look at fixed cells or live cells. So the BioPipeline series from Nikon allows you to look at fixed cells on plates, that's shown here on the left, or you can use the BioPipeline Live to look at living cells with a full incubation and environmental control around the microscope, and turnkey automation that will transfer, no matter what the storage compartment is, cells and samples from that storage compartment onto the stage for an imaging experiment. So you can do cell-based screening on the phenotypic side, on the simpler side, with something like this. This is an example from Steve Finkbeiner's lab at UCSF and the Gladstone Institutes. And this is a pretty simple readout. This is a binary readout, red, green, how much of each. And it allows you to, you can seed these cells that report either red or green based on the conditions that have been determined for the experiment. And depending on how they've been treated, they either have more green or more red, right? A pretty simple readout. And this allows you to do the fixed time point imaging and quickly assess the effect of your compound on the cells in culture. And you can do this in a 386 well plate, or a 384 well plate, or a 96 well plate. And so the throughput is very high, and the readout is very simple. And at the end, you just analyze with a heat map the amount of green or red in your sample, right? So this is the simplest type, just a quick fixed readout where at the end we can quantify green and red. Then the next level of complexity is something that's been around for a long time. It's something we call multiplexing, where we actually, and this is more along the lines of what Salomemix was doing to generate that 10 to the 8th number of parameters. But what you do is you image the cells at a higher magnification so that you can see features of the cell itself and then the organelles within the cell. And this combination of high magnification along with high content, where we're imaging many fields of view at a time in many wells, allows then for the researchers who are doing this to create a library of cell images for a given phenotype. And then these can be interrogated over and over again, depending on the question you want to ask. It's nowadays they call it cell paint. This is an example of what that looks like from the Broad Institute at MIT. And this is something that has gained a lot of popularity and something we see quite often now, where you will take high resolution images, high magnification images of cells, and then create masks. So you start with the cells raw data here, and then you look at different features and create masks that then quantify those features, and then you can compare different conditions using these features. So this is a slightly more complex version of that technique of doing a fixed time point phenotypic screening. So what if we add the live component? Then we can get even more information. So this is actually an example of research that was done using a live imaging system, and it gives you a more nuanced view of how a compound might be working. So in this experiment, the cells are kept in culture and they're imaged over time. And so at the 0 hour mark, then a compound is added. And in this case, the cells are neurons, human neurons that have been derived from human iPS cells. And so the relevance is high because these are actually human cells. And the goal is to understand how they're affected by the treatment. And these cells are the control groups include a healthy group and then a group that's been damaged somehow to represent neurodegeneration, right? And then compounds are added to the injured groups to see which compounds reverse the neurodegeneration, or in other words, lead to more healthy cells by the end of the experiment. Now, if we were using a more simple phenotypic approach, then typically what we would do is we would treat the injured cells at time 0 with the compound or compounds that we would hope would be able to restore function to those neurons. And then at some time point T, we would stop the experiment and fix the cells and then image them and count how many cells are alive and how many cells are dead. But what if there's information we're missing between 0 hours and 120 hours that we'd like to have so that we can understand the mechanism of that compound and how it's affecting the health of the cells at the end of the experiment? Or in other words, why is it that we get more live cells with certain compounds than others? And that question is difficult to answer, in fact, impossible if you're only doing a fixed time point assay. So you have to create an assay that's robust enough to deal with cells, and you treat each cell almost as an individual. And then you can extract a lot of information over time about how this compound is affecting the cells that are being treated. And so shown here is kind of a graphic that indicates at time 0, you have your cells. Each one has a number of nodes. That's what NON stands for. And we can actually label these nodes using software analysis. And over the course of the experiment, cells become less healthy or more healthy. You even have cells that show up, like the cell E, and the system can handle this kind of fluctuation in the population. And at the end, then what we can do is we can monitor over time what these nodes are doing, the number of nodes that we have, and use that as a surrogate for the health of the cells in the culture. So what this looks like in real life is something like this. And the cells that are tagged in green have been determined to be healthy. The cells that are tagged in yellow are determined to be unhealthy. And then the cells that are tagged in red are dead. And so over the course of the entire imaging experiment, we can track cells that start out unhealthy. You can see they start out yellow, but then they become healthy, they become green, right? Or cells that might start out yellow and become red over time, or cells that were red and they stayed red. This allows a more nuanced approach that then we can say a certain population of cells started out sick and they became well, or a certain population of cells started out healthy and they stayed healthy. And we can start to make distinctions about mechanism of action for different compounds. And in fact, we can do that, we can quantify that. Shown here is an example of that quantification where we have healthy cells, unhealthy cells, and dead cells, right? And if we add a drug that maintains the population of healthy cells throughout, then we know that's a neuroprotective drug, right? And it may not be a good drug to give to people who are already sick, but maybe it could be part of a treatment regimen, prophylactic treatment regimen for people who are at risk, right? And then other compounds might take unhealthy cells and make them healthy again. We can see this in this type of an assay where we couldn't with the fixed time point assay. Those types of drugs we would give to people who are sick, right? And both of those drugs look exactly the same with an endpoint analysis if you're not watching the behavior of the cells over time. So this is a powerful example in the neuroscience space of how we can use kinetic imaging and modern image analysis to gain mechanistic insight on a high content screening level. So, this is kind of the pinnacle of phenotypic screening, I would say, where you get the most information, both spatial but temporal information as well. And, obviously this generates a lot of data. So there needs to be tools that match the hardware in terms of handling large amounts of data that are being produced. And so what's nice is modern tools include the integration of artificial intelligence. And these allow us to do things like label-free imaging and what I would call like a digital staining, right? Where you have here a nice high contrast image that's been collected in phase contrast, and then you can add on top of that a pseudo stain. Now these cells haven't been labeled, but we can mark their cell boundaries, we can mark their nuclei, so we can count the number of cells, we can look at the displacement of the cells over time using these AI tools. So now, for instance, if we don't have a marker that will give us the phenotype of interest, we can just say the cell morphology is our phenotype and do no staining and kind of draw conclusions based on observations using these techniques. We can also eliminate background using artificial intelligence. One of the problems when you're doing these large screens is sometimes there are wells or samples that have poor signal to noise. And in this case, it would be very difficult to pick out this kind of blue background from the actual cells and even do something as simple as cell counting. But modern AI is very good at finding patterns and removing patterns that we don't want and keeping the ones we do. And so we can actually remove this background and create a very nice signal to noise, and we can go so far as to count the numbers of cells, but we can also look at their shape and we can extract those features like the Salomemix example showed, and we can extract hundreds of features from even what used to be unusable data, right? And then finally, we can look at things that are somewhat complex, like branching and morphology. This would be difficult to do without the help of an artificial intelligence software tool, because while we can see as humans there are all these branches on these neurons, for instance, it's difficult for a software tool to do the same thing using conventional methods, or for a human to show the software how this is done. But using an AI, like I said, that's very good at finding patterns, we can train an AI, this is what a projection from a neuron looks like, and then it can be very good at finding these very, very quickly, and then quantifying the number, the length, the number of interactions between neurons. And so this is another example of how artificial intelligence powered software tools are helping advance the industry and advance the science. So that's on the tool side, but what about on the model side? So there are some very nice new 3D cell models that are coming out that allow us to use human cells arranged in 3 dimensions to give us a more high fidelity model that takes us away from having to use an animal because it gives us some of that 3D structure and context. And so we get away from the animals a little bit, but also we can maintain the strengths of kind of the in vitro system by being able to do many of these, right? So some example are organoids, which are 3D arrangements of cells, and organs on chips, which is a microfluidic device that has very high control over the spatial arrangement of different types of cells. So you can create things like lungs or kidneys or livers or colons, these kinds of things in a chip that can then be used for screening. Some very nice examples from Deckers et al. from a Nature Protocols paper show the power of these organoids. And we can see different organoids here shown across the top. And then if you zoom in, you can get very high quality detail using a nice confocal microscopy system. And so this shows that now we can get microstructures that are very reminiscent of physiology in a screening context where they're in a dish or a well, and they're very accessible for doing both low and high magnification imaging. And then we can quantify these things. This is an example of the organs on a chip. This is a system from a company called Emulate. And this can actually take endothelial cells, human endothelial cells, can be seated in one channel of this chip. And then there's a membrane, a semi-permeable membrane. And then we have some functional cells, in this case, proximal tubule cells. And this creates kind of a kidney microenvironment. And so compounds can be flushed through this vascular compartment here. And then as they diffuse through the membrane and hit the functional cells, then function can be assessed. We can look at things like metabolism, or we can also image these things. So using a high frame rate confocal microscope, we can image through hundreds of microns of this model system very quickly. And this is an example of what that looks like. So we start out in this video set going from the basement membrane. So what we're going to do is we're going to go, or the basement of this luminal compartment here. So we're going to start here and we're going to move through. It's going to get dark for a bit, and then we're going to hit the second layer of endothelial cells, and then we're going to hit the tubule epithelial cells, the proximal tubule epithelial cells in this image. So we start out at the base, we go through the luminal compartment, and we can see the cells crawling up the sides here. And then we get to the other side. Now we can image the other set of endothelial cells and now the proximal tubule cells. And we can actually make out the tubule structures in here very nicely. So now we can combine these high-end three-dimensional human cell models with imaging to get a complete picture of what's happening in this system based on either, like we've said before, genetic differences between sample types in the cells, or by treating them with compounds. And we can get a very nice high resolution image from these that can then be used for things like 3D modeling, where we actually look at the structures of the components, for instance, the proximal tubule structures under the influence of different genetic and chemical perturbations. So again, this is all very exciting. And these are some examples of how we can use modern imaging techniques, hardware and software, to screen in modern screening models. So let's switch gears for the next few minutes and just talk about how this is now being applied in the area of therapeutics research and development. So especially in an age where there are a lot of therapeutics coming out that are cell-based, then FDA has signaled they're willing to accept morphology as a criteria for creating quality control measures that are used for getting approval for your therapeutic, but it needs to be quantitative, right? And there are a lot of things that imaging can do that is difficult to get from flow cytometry. And I'll show you some examples. So in the first case, iPSCs, or induced pluripotent stem cells, are a rich source for all kinds of tissues or cells that can be used therapeutically, but they have to be generated first. And so how do we tell in an automated fashion the difference between an actual iPS colony and a non-iPS colony? Even for a human, this is difficult. But we worked with Shinya Yamanaka at CIRA to figure out, is there a way to do this in an automated fashion? And in fact, there is. So we can create image analysis routines that look at the emergence of these iPS cells as they're being reprogrammed, and we can quantify the morphology to the point where we can tell what's a real iPS cell and a non-iPS cell, and we can determine the efficiency of reprogramming, and we can identify individual colonies that should be removed and used for further expansion. So that's at the iPS derivation stage. Then once you have them, you need to assess their health. So over time, you have colonies that grow, some become differentiated, some batches become so differentiated that they're not useful anymore and they need to be discarded. And we need to be able to, in an automated fashion, track this. And it's difficult to do this in line with the production process using something like flow cytometry. But with microscopy, we can actually watch all of our samples as they're growing, all of our batches as they're growing, and then we can tag those with identifiers. Shown here in different colors are each of the colonies that are being generated during the culture process. And then you can quantify their health over time by looking at parameters such as size, morphology, these kinds of things. Shown here in the graph on the right. And we can even take this further and we can tell which ones have been differentiated and which ones, where the differentiation is and where the pluripotent cells remain. And if we have too much differentiation, we can flag those for removal. And the fidelity is quite high. So if we compare this kind of pseudo staining where it's label free, there's no fluorescent markers or dyes or proteins. This is just the software has identified the differentiated area from the undifferentiated area. We can see that the fidelity with immunofluorescence, which is kind of the gold standard up until now, is quite high. And we can take this even further. We can look at differentiation. So this is an example now of creating a therapeutic where the therapeutic is a neuronal progenitor cell, right? And we're going from human pluripotent stem cells through a differentiation process. And we want to make sure that during the differentiation process, we're getting what we're supposed to be getting so that our progenitor cells have the expected behavior and phenotype. So what we can do is we can use software to create masks that identify different populations of cells, each one representing an important progenitor population that gives rise to the next. They move in kind of a stepwise fashion. And as long as this progression is observed, then we know that the product is the desired therapeutic product. And so we have kind of this orange, or in the end, we'll have the kind of purple, pink, and green cell clusters that we can track over time. And again, an example of the actual phase data, and then the digital staining that's done with the software where it can recognize different cell populations based on their morphology and their behavior. And then kind of a confirmation, this is the control where it was then stained. And we can see that over the course of the differentiation experiment, then we have our populations remain kind of stable as the first progenitor population grows, shown here in the purple. And then once that reaches a steady state, then the green population starts to grow. And then that reaches a steady state and the pink population grows. So each one gives rise to the next in true developmental fashion. And now we can use this method, this imaging method over time to show that our cells have the proper identity and that they're ready for either the next stage of differentiation or they're ready for therapeutics. If we don't see this nice progression, then we might assume that this batch is bad and we drop it before it gets to, the 2, 3, 4 week period, and then we find out that it's bad. Why don't we drop it earlier in the production process before we wasted all of that time, right? So this is a way where imaging allows us to look at the cell product that's being made and then have some go, no-go criteria that allow us to make decisions early on in the process. This is an example of looking at epithelial to mesenchymal transition. So this would be, quantifying an important process for like a vascular therapeutic. Or looking at the health of neurons after the differentiation has completely occurred to make sure that they're ready for either use as a tool or as a therapeutic. And again, this is without any labels. This is completely label-free. We can look at multiple parameters over time and assess the health and viability of these cells to be used as a therapeutic. So that's about it. So, you can see how powerful imaging is, and when it's applied both in research and development, which I think is a more traditional context, um, and when it's applied after that for screening of especially these more complicated therapeutics like cells, then there are a lot of modern tools both on the hardware and software side that allow us to extract quantifiable information. So you can see why I'm so excited about this. I think this is really an amazing thing, and you can create lots, large libraries of data that can be interrogated over and over again in different ways to give you different information. And it can be used for, regulatory approval on your process, right? Imaging, in fact, in manufacturing has been done for a long time. Semiconductor industry uses it to look at their products and make inline decisions about the process. And Nikon even does this for that industry, and it's moving over now into the biology side as well. So it's exciting to see some of this crossover in what's happening. But as a general summary, we can see that observing the biological responses to environmental stimuli is what makes up these screens, and that this is really a bedrock in life sciences discovery. The phenotypic screens in vivo and in vitro allow us to see from the subcellular level all the way up to the organ level what's happening. And when we add a compound, or we have a genetic variant, or we introduce a biological or a cell therapy product, and then modern technology allows us to do this in a way that really moves the industry forward at a pretty high pace, but also once you've gotten past R&D into the production phase. A minute to talk about what Nikon can do about it. On the imaging side, I've talked about the Bio Pipeline series. This is a new series from Nikon that provides a simple software interface with integrated artificial intelligence and integrated environmental controls and robotics for handling of samples. It also includes a server for high content data handling and storage. This is really exciting because with one turnkey solution, you essentially have an entire automated phenotypic screening lab right there on a table, right? It's really incredible, and it's kind of the culmination of all of these techniques and technologies. I'd also like to share a little bit about another product that's come out from Nikon recently, and this is a contract research product. This is where we do the research for you. A lot of times equipment can be expensive to buy and maintain, and it's also challenging to find the expertise in terms of personnel and hire people who know how to use these instruments in the way that will give you the best data. And so in talking to a lot of biotech companies, a lot of startups, even large companies, what we found is that there's a need for contracted work to be done that allows the collection of microscopy data that's mission critical for a lot of companies. And so that's where we've created the Nikon Bio Imaging Lab. And at the Nikon Bio Imaging Lab, we have staff who can handle any type of experiment along the lines of the ones that I've shown you in this PowerPoint, including screening and 3D imaging. And we can, do anything from you just send us your samples, we image them and send you the data, to send us a protocol, we culture the cells, we run the experiment, we do the data analysis and send you back quantitative data as well as imaging data. But we're excited to offer this to the community, and we do it in a very simple contract rate. And so, whether your budget is very small on the order of hundreds of dollars, or if it's large on the order of hundreds of thousands of dollars, we can scale our services to fit your needs. So we're excited to offer this to the community. And that's it. So thank you very much for your attention. I'm glad that you could join me today. Like I said, I'm excited about this, and I'm always excited to hear from people who are in the field, who want to learn more, or with whom we can work to provide solutions. So please feel free to reach out.
Hey, Jeff, we do have a couple questions for you just to wrap stuff up. First of all, one of the questions that popped up was, which microscope is used for the kidney chip imaging?
Yeah, I'm glad you asked. Actually, that was something I did on a Nikon confocal microscope. It's our A1R HD HD25 microscope with a special objective. It's a 20x objective that has a very long working distance. It's about 2.5 millimeters of working distance, but the numerical aperture is very high. And so it allows us to get really high resolution data very fast. So I was able to get through that entire chip from bottom to top in a matter of about 20 minutes, which, you know, the entire chip, which is unheard of, right? But our software, our hardware are designed for this kind of high speed, high resolution imaging. So that was the A1R HD confocal. Thanks, Peter.
I have another one that's just popped up. It says, where do you have such contract services? Is it in the Bay Area, San Diego? Could you please clarify?
Well, we're based, our lab is based in Boston, but we accept contracts from everywhere. You know, it's kind of nice. I think we've seen, especially with this, you know, pandemic situation, that a lot of things can be done remotely. And we've worked with customers across the country. They'll send us samples or they'll send us protocols and we'll run the services. But our lab, our physical lab location is right now based in Cambridge, Massachusetts, just across the river from Boston.
Okay, I have another one. Could you elaborate a little bit more on the advantages of using imaging for quality assurance?
Yeah, definitely. So in the case of quality assurance, you know, I could elaborate for hours, but what I'll say is that, you know, a picture is truly worth 1,000 words, right? And it's worth 1,000 biochemical assays, I would say, in this case. So if you're looking at a system that includes a living, you know, that's alive, right? Then you can get a lot of information. So in the context of, like I said, looking at cells. There's a lot of cell therapy happening right now. Then when you collect an image, then you are getting data that includes spatial information, relative, relative positional information amongst different individuals within your sample that is beyond what you would get from another, say, like an enzymatic activity assay. And because of modern software, we can extract a lot of information that would tell us about what's going on, even on the molecular level, by just looking at the phenotype. And, you know, I think once you've collected that data, then you may have some kind of criteria that you've created, like a CQA, right, that you're using to evaluate your product. But let's say that 6 months later, you realize, hey, there's something else we should be looking for. You have those imaging data already. So you don't need to go back and rerun everything again. You can just pull those imaging data and you can look at them and you can say, oh, hey, if I reapply my algorithm in this way, I can get more information along the lines of what I'm interested in now that I may not have been aware of 6 months ago. The other thing that's nice about it is that if you're taking pictures at a regular interval, then let's say there's a problem. And at any point of the production process, then you can say, oh shoot, you know, we had contamination, but we're not sure where it came from. Well, you go back and look at your images. Your images will tell you, oh, this is where it happened. We can start to see the infiltration of bacteria here, or we can see a color change happening here that we didn't notice at first, but now we can say for sure, yeah, this is where it started. And then you can use that to evaluate your process and see what needs to be changed. Right, so there's a lot of examples where imaging data is kind of not only useful now, but it's a little bit future-proof, and it provides a built-in kind of a log, right, of data that is required by most regulatory agencies anyway. So you've already got that built in if you're taking images. So it's really nice.
So Jeff, I think we have time for just one more question, but for those who are still on the call, I want to also make sure the Nikon program does have special pricing for bio and state affiliate members. So if you are interested, understand there is significant discounts for these systems available to you. Okay, one last question here. Relative to the advancement of new technologies coming up in the near term or future term and how it might impact the industry.
Yeah, great. Thank you. So I think, you know, to the point about that Peter made about contract discounts, we're excited to offer these both on our instrumentation and also on our services to bio members across the country. And so please reach out and ask us about our bio rates. We even offer free pilot experiments for the contracted research. So thank you for bringing that up, Peter. We look forward to hearing from your members about that. And then also, when it comes to the future technology, there are a few things that I'm excited about. One of these that I've touched on a little bit in the presentation is artificial intelligence. There are some really amazing things that we're doing in this area where we take advantage of this pattern recognition, right? To really create processes that can be carried out, or imaging analysis techniques that can be carried out in line, meaning that you can monitor the product you're making and extract information from it throughout the production process without having to take a sample off and then do some kind of other analysis, right? Or on the R&D side, now we can run multiple, hundreds of plates of, say, organoids, right? And we can image through them quickly because of some artificial intelligence integration that removes noise. But then we can do quantification on patterns that were previously inaccessible because of the amount of time that would take for a human to go through and make these threshold adjustments. But by doing an iterative process with an artificial intelligence, then we can automate all these processes in a way that we've all been anticipating for a long time. And then the other side of that is hardware, optics, right? So Nikon, in addition to being experts with the software, we're also optical experts. And we have all kinds of imaging systems coming out, taking advantage of nice integration between really high-end optical solutions in the glass side, for instance, but also integration with hardware that takes advantage of this. And so I can see in the future a really nice interplay between the AI, the advanced optics, the advanced hardware, kind of automation that's turnkey so that people of all types can access this automation is another thing that I see that's very exciting. And again, this is another thing I could talk about for a long time, but I'll leave it there. So thank you very much, Peter. I appreciate it.
Yeah, and for those participating, we really do appreciate your time. I know it's the top of the hour and everybody is busy. So again, here's Jeff's contact information. And if you have any questions, please don't hesitate to reach out. Thank you again for your participation, the questions that were raised today, and everybody have a great and safe day. Thank you very much.
Thank you, Peter. It's been a pleasure. Appreciate it.